we uncover the genetic connections between Djibouti, Africa, and Asia. Our analysis reveals how ancient migrations shaped Djiboutian DNA, highlighting intermarriage, trade, and cultural exchange. Explore the Cushitic heritage and Arabian influences that formed this unique genetic tapestry. With each genetic marker uncovered, we move closer to understanding how Africa and Asia were never isolated worlds, but intertwined for millennia through shared bloodlines, marriages, and cultural exchanges. Djibouti stood at the heart of this biological dialogue, a liminal space where the continents whispered across the water. Modern Djiboutians, in their genetic makeup, carry the legacy of this crossing. They are living testaments to the movement of ancient peoples, a bridge made not of stone or steel, but of chromosomes and mitochondria. Their very existence defies rigid classification and reminds us that the story of humanity is not about separation, but about connection. In the unfolding story of our species, Djibouti's genes offer a crucial chapter, not a fringe or an afterthought, but a centerpiece in the genetic architecture of the Afro-Asian world. As new studies delve deeper into this bridge of DNA, they uncover not just migrations, but a symphony of survival, interaction, and human ingenuity that shaped the biological foundation of two continents. In the complex genetic mosaic of Africa, the Cushitic-speaking populations of the Horn represent one of the oldest and most enigmatic threads. Djibouti's dominant ethnic groups, the Afar and the Issa Somali, speak Cushitic languages, which belong to the larger Afro-Asiatic family. But linguistic ties are only part of the story. Beneath the spoken words lie genetic clues that suggest ancient origins and unique evolutionary paths. Cushitic languages are among the oldest living tongues in Africa, some with features traceable to the pre-agricultural era. This linguistic longevity is paralleled by genetic continuity. Genome sequencing of Djiboutian individuals, especially from inland regions less affected by recent migration, shows preservation of ancient African haplogroups that diverged tens of thousands of years ago from other African and Eurasian lineages. Y chromosome analyses among Djibouti's Cushitic speakers reveal a significant presence of haplogroup EM78, an ancient lineage thought to have originated in northeast Africa. This haplogroup is often associated with early pastoralist migrations and is found in high frequency among Cushitic speakers throughout the Horn. Its consistent presence across generations in Djibouti suggests long-term regional stability, groups who settled early and remained rooted. But the story deepens when we examine mitochondrial DNA, especially lineages such as L3 and M1. These maternal haplogroups are like ancient threads spun through both African and Eurasian populations. L3, for instance, is thought to be the mother of all non-African maternal lines, and its roots can be traced back to East Africa. M1, on the other hand, is a genetic echo of back migration from Eurasia into Africa. Likely entering the Horn through Djibouti thousands of years ago, the unique blend of these haplogroups in Djiboutian Cushitic speakers points to a dual ancestry, ancient African roots interwoven with early Eurasian contact. This intertwining doesn't imply invasion or colonization, but rather slow, complex processes of intermarriage, trade, and cultural exchange. The Cushitic code embedded in the DNA of Djiboutians reveals a population that acted as both hosts and wayfarers in human history. What's especially intriguing is how this genetic signature aligns with archaeological findings. Pastoralist sites in the region dating back 6,000 to 8,000 years show evidence of early cattle domestication and obsidian tool use. These early herding cultures likely moved in seasonal rhythms, following grazing lands across Djibouti's arid plains. Their movements left not only material traces, but also genetic legacies that persist in today's Cushitic speakers. Recent advances in genome-wide association studies, GWAS, have allowed scientists to identify specific traits linked to Cushitic ancestry. For example, certain genes associated with lactose tolerance uncommon in most African populations, appear at relatively high frequencies in Djibouti. This adaptation likely reflects the pastoralist heritage of Cushitic groups who relied on milk as a critical food source in dry environments. Skin pigmentation genes also show unique variations among Cushitic-speaking Djiboutians. While most African populations carry the darkest pigmentation alleles, some Djiboutian groups display intermediate shades. Geneticists have found that this is not due to recent European admixture, but may represent ancient diversity within Africa itself. Another indication that Djibouti preserves early human variation, rather than being a product of modern mixing. The Cushitic genetic code also preserves adaptations to aridity and heat stress. Genes affecting sweat gland function, salt retention, and metabolic water usage appear in patterns that differ from those seen in tropical or highland populations. These are subtle biological features evolved over millennia to survive in Djibouti's semi-desert terrain. Once again, it is the continuity of life in one region, not rapid movement, that tells the story here. Further complexity arises when we look at the relationship between Cushitic and other Afro-Asiatic populations, such as Semitic speakers in the Arabian Peninsula and North Africa. Despite the shared linguistic ancestry, genetic data reveals marked differences. Djiboutian Cushitic speakers show minimal overlap with North African Berbers or Levantine Arabs, suggesting that the Cushitic genetic profile developed largely in isolation from these northern groups. This supports a theory that the Cushitic-speaking ancestors were among the earliest Afro-Asiatic groups to differentiate, long before Semitic languages spread from the Middle East. Thus, Djibouti becomes a genetic reservoir of an ancient population that may have diverged 10,000 to 15,000 years ago. While their language shares a family tree with others, their DNA roots dig deeper and in a different direction. The Cushitic genetic legacy isn't just a relic of the past. It actively shapes the health and resilience of present-day populations. As geneticists explore disease prevalence and pharmacogenomics in the Horn of Africa, understanding the Cushitic code becomes increasingly vital. Genes related to immune response, glucose regulation, and kidney function are being studied to tailor medical treatments and anticipate vulnerabilities specific to this population. Even in the age of globalization, the Cushitic DNA of Djibouti remains remarkably intact. It is a silent library of ancient adaptations, migrations, and interactions. Every gene sequence tells a story not just of survival, but of identity. A narrative carried not in books or monuments, but in blood, bone, and breath. In decoding the Cushitic genome, we are not merely retracing history. We are discovering how language, landscape, and lineage converge to form a people uniquely adapted to their world, yet deeply connected to the larger human journey. Djibouti's Cushitic legacy stands as a genetic time capsule, one that guards the ancestral rhythms of Africa's northeastern frontier. Across the shimmering waters of the Red Sea lies the Arabian Peninsula, a land that has long shared invisible threads with Djibouti. Though today divided by the Bab el-Mandeb Strait, the genetic, cultural, and spiritual connections between these two lands stretch back millennia. Among the most enduring of these are the traces of Arabian ancestry hidden within the DNA of modern Djiboutians. For centuries, if not millennia, Djibouti and southern Arabia maintained a relationship built on migration, trade, and intermarriage. This connection was not incidental. It was driven by geography. The narrow strait, often navigable even with the simplest sailing technologies, enabled people to cross in both directions. Genetic evidence now suggests that these crossings were not occasional, but sustained and influential. Genomic sequencing in Djibouti reveals the presence of haplogroup J, particularly J1, in a portion of the population. This paternal lineage is common in the Arabian Peninsula and parts of the Levant. Its presence in Djibouti cannot be easily explained by recent contact alone. It likely represents ancient admixture events that began during the Neolithic or early Bronze Age. These were eras when domesticated animals and farming knowledge also began to cross the Red Sea. Interestingly, mitochondrial DNA studies also show the presence of haplogroups that originated or expanded within the Arabian Peninsula. This confirms that the genetic exchanges were not limited to male-dominated merchant or pastoralist migrations. Women too were part of these movements, whether through marriage, settlement, or joint community migrations. The result is a Djiboutian gene pool with subtle but consistent Arabian signatures. Archaeological evidence complements these findings. Excavations along Djibouti's coast, particularly around Obok and Tajura, have uncovered fragments of South Arabian pottery and obsidian tools sourced from Yemen. These objects are not merely evidence of trade. They suggest settlement, interaction, and shared life ways between communities on both sides of the sea. These exchanges may have intensified during the Sabaean era, roughly 3,000 years ago, when South Arabian kingdoms flourished and established trade networks that included the Horn of Africa. These maritime routes were not just commercial. They were conduits for genes, ideas, and culture. The genetic influence of these South Arabian settlers remains detectable today in specific autosomal DNA segments within Djiboutian families. Another key moment of genetic fusion came with the rise of Islam in the 7th century CE. As Islam spread from Mecca and Medina, it followed both land and sea routes into Africa. Djibouti, situated along a major maritime artery, became one of the earliest African regions to adopt the new faith. With it came an influx of religious teachers, traders, and families from Yemen, Oman, and the broader Islamic world. Genetic analyses of the Djibouti and population during this historical period show a slight uptick in Arabian ancestry, consistent with migration waves accompanying religious and social transformation. However, this was not a one-sided absorption. The Arabian newcomers also integrated into existing African communities, creating a genetic dialogue rather than a replacement. One intriguing marker of Arabian contact is the presence of the Duffinal allele among some Djiboutians. While common in sub-Saharan Africa, its moderate frequency in Djibouti, alongside Arabian Y DNA lineages, suggests gene flow from both regions and local adaptation to malaria environments. This convergence is a reminder that genes are not static. They are constantly shaped by geography, environment, and intermarriage.